All right. Hello, everybody. So we are having to do this online because weather has made it so that we cannot meet in person, unfortunately. I don't get to see you guys very much, and now we have an entire session that has been taken away from us. This slide is just kind of a heads up of everything that you have going on right now, all of your due dates that are coming up in the meantime. So we do have your homework 3. So your homework 3 is due February 21st. This is Sunday by 11 p.m. There is a section on Blackboard for you to turn this in. The instructions are also in Lab 3. It's the very last page, I think, of Lab 3. So if you want more, I will talk about it more at the very end of this lab, but make sure you do this. That slip of paper that I gave you that says MHC and it has a topic with it, that is your topic. That's what you should search. If you don't remember what it was, you didn't get one, whatever, use one of these 3. Don't do all 3 of them, just do one. Anatomy, sound, or habitat. Pick one and do that, okay? If you have questions, let me know, but do not email me at 10 o'clock on Sunday night because I'll be asleep. Next, so you do have a long-term experimental design worksheet, and this is going to be due Friday, February 26th at 11 p.m. So you're going to have to email this to me. You're going to find out, I'll talk to you guys about this a lot more at the very end of this video. I have an example for you guys. The example is on Blackboard. The whole thing is on Blackboard for you guys. You're going to email this to me, and this is a group effort. So you're going to work with your lab partner. This is where it comes in handy that you guys got your lab partner's information. If you didn't, or if you can't find it, then let me know, and I'll pair you guys together and give you guys your emails. But this is I'll talk about this way more at the end. The next thing, a lot of you have already done this, so I shouldn't have many left, but go ahead and turn your preliminary exercises into me by email. I'd really prefer that you do this by Friday, which is the 19th, so tomorrow, by 11 p.m., okay? So you guys have all day to do that. They should already be finished anyway. Just take a picture, send them in. If you're stuck somewhere, then let me know, and we'll get things figured out. The last thing, your quiz 2, so you have a quiz for Lab 2, and obviously we can't do it because we didn't meet in person, so instead, you're going to turn it into me, so we're going to actually do this at the very beginning of the next class, okay? So I will remind you of this so that you guys don't forget and come in completely blindsided that you have to take a quiz. But just as a heads up, this is something that you guys are going to have to do the next time we meet, okay? So you have quiz 2 at the beginning of lab, and then you'll have quiz 4 at the end of lab, okay? So just prepare for that. Again, if you have any questions about any of these, please email me, okay? All right, so we're going to go ahead and get started with our actual PowerPoint and everything. So as always, we start out with a fun little thing for you guys to look at. This is just a cartoon. It says, it has a wasp with 2 cockroaches, and it says, first of all, we want our name changed from cockroach to companion beetle. And this is a really funny thing because most cockroaches are disgusting and we don't want them as companion beetles. However, our lovely Madagascar hissing cockroaches are greatly loved, right? So it's just a little funny. Alrighty, so getting into all of our stuff. So this class, we're looking a lot at science and experimental design and that sort of thing. So science, as we've kind of figured out, is a very broad term, right? This is a really broad spectrum type of thing. Science, as is discussed in your book, is going to be a way to explain the natural world, right? So we're going to use science as a way to explain everything that goes on around us, essentially. Within science, there are many different disciplines, right? So looking at science, it's a very broad term and within that you also have physics, right? Which is going to be understanding forces. You have chemistry, which is understanding matter, and you have geology, which is going to be understanding the composition and changes of Earth. All of these are different types of sciences. And within this, you also have biology, right? Which is the study of life. However, you can go and break it down even further. So within biology, we have various other subdisciplines, right? So we have zoology, which is kind of what this class is, which zoology is the study of animals. You also have the study of plants, which is going to be botany. You have the study of bacteria and fungi, which is going to be microbiology. You also have my personal favorite, which is entomology, the study of bugs. So there are lots of different things that you can look at when it comes to science. There's a lot of different areas within science. Some of you are in animal science, right? So this is sort of it's a variation of zoology in many ways, right? So lots of different ways that you can look at science. So science is a way of knowing and learning. So science is very evidence-based, right? We know this. Science is evidence-based. We have concrete evidence that we find in order for it to be considered science. Science is typically based off of data and particularly numbers and something that you can do over again if necessary and to figure out if it is actually correct. Within this, we also have religion. So many of you guys probably are religious. And within religion, you know that it's very belief-based, right? It's not, you can't see it, you can't touch it, but you believe that something is there. You believe in God, or you believe in this. Typically, the two don't come together, and that's because one is evidence-based and one is belief-based. We keep them separate as often as possible, but sometimes they do come together, and this occurs rarely, but when it does, it's typically for the greater good of others, and it's more for rules and regulations. So an example of this is doctors. So when a person becomes a physician, they have to state that they will not harm patients. They will not kill their patients. And this is sort of based off of religion, right? But for the most part, we keep science and religion completely separate. It is completely possible to be religious and a scientist. There are many people in this department that are of every religion you can think of and still great scientists. It's just one of those things that you have to make sure you know the difference. Science is evidence-based, religion is belief-based. So, like I said, science is very evidence-based. We need this. Science is tentative because of this. Evidence can change, and it changes mostly because of technology. So the more technology changes and evolves, the more our understanding of a specific subject is going to also change and evolve. And there are many different examples of this, but one that you should think of that's going on in real time right now is everything with COVID. So when we started with COVID-19 almost a year ago now, we didn't know a whole lot about it. There were a lot of articles being published about it. And while none of these articles were completely correct, none of them were wrong either. And the reason this is because the technology changed. Our understanding changed because we were able to look at something better or look at something longer. And so science is constantly changing. Another example of technology changing is your cell phones. How many of you guys probably had a flip phone growing up? I know my first phone was a flip phone. So, I mean, that's completely different than our incredible smartphones that we all have now that can go on the internet and everything else. I remember on one of my phones that I had that was not a smartphone, if you touched the internet button, you were screwed because it wasn't going anywhere and you just got a whole bunch of data stuff that was not okay. So, you know, it's always one of those things. Technology is constantly changing, and for the most part, it's pretty cool. So, a really good example of science changing and because of technology is the center of our solar system. So, originally, we believed that the Earth was the center of our solar system, right? So, we thought this for a really long time, and after a while, we figured out that the sun is actually the center of our solar system. And so in your books, we do talk about this a good bit. We talk about how this happened and how we figured this out. The main way that we figured this out was because of evidence found by Nicholas Copernicus. So Nicholas Copernicus was a brilliant mathematician that came up with a mathematical equation that showed that the sun was actually the center and not the Earth. Now, when he came up with this, he was essentially the only one that could understand this math involved. He was the elite of the elite when it came to math. And so everyone thought he was crazy because nobody could understand it. Until one day, This other guy came along named Galileo, and he invented the telescope. And when he invented the telescope, he was able to prove that what Copernicus had done with the math was actually correct. The sun was indeed the center of our solar system. And he did this because the technology changed. He advanced the technology to give evidence that Copernicus's math was correct. Pretty neat thing, right? So, here we have the scientific method. This is just something I have on here for you guys. I talked about it a lot in my other video. But if you have more questions about the scientific method, please let me know. We use a very cut and dry scientific method in this class for you guys. We try and make it as simple as possible for you all because you do have to do this for your oral report, right? This is something you need to understand the whole process of. However, there are some instances where the scientific method isn't always used quite like this. One really great example of this is going to be Alexander Fleming. So, everyone's probably heard of Alexander Fleming. He is in your book, at the very least. So, he's actually the man that discovered penicillin. Penicillin is an antibiotic that we use today still. So, it's pretty awesome, right, that he discovered this. And so the way he discovered this was because this man had an absolutely disgusting lab. He would leave petri dishes all over the place and never really pick stuff up. And so one day he noticed that a petri dish was growing something in the middle, and all around this petri dish, this piece that was growing in the middle, it was pretty clear. There was nothing else in there. And because of this, he figured out that he'd actually created an antibiotic on accident. And so, here you have penicillin. Very awesome. But typically, we do not come by our scientific discoveries on accident. They are found through the scientific method. One thing that is super important for you guys to understand is going to be the peer review process. So, peer review is essentially how scientists get their information out to the world so that everybody else can understand it and see what is going on. A peer-reviewed article is how you're going to know if an article you're reading or anything you're reading is based on scientific fact. It's not from like a random person that lives in the basement with no scientific degree, right? This is how you're going to figure this out, is through a peer review process. So a scientist does a whole experiment and they come up with these awesome results, and so they write a paper on this and they submit it to an article that they, or a paper that they think is going to do great. So it goes to an editor, and then if it passes that, then it goes to reviewers. And so these reviewers are going to be experts in this field. Typically, they're made up of peers. So within fields, particularly those that are fairly small, you're going to know all of the people that are in that field. And so that's one of the reasons it's called peer review. It's people that are also in this field that are experts as well. And so they're going to assess this, and they're going to figure out if what you've done is correct, and if it can be done again, and if make sure you're not falsifying your results. So this is one of the biggest things really that is important to figure out if you're looking at a actual scientific article or not. Unfortunately, it does happen sometimes that someone will perform scientific misconduct. This does happen every once in a while, and it's very unfortunate. One of the most widely known cases that you guys probably all do know about is a physician in the UK created a study to see if vaccinations cause autism. And when he did this, he actually falsified and changed up his data. And so his paper was published, and it caused a whole lot of issues with people getting their children vaccinated. And so turns out everything in that was falsified and none of it was true. And so because of that, you ended up with a huge anti-vaxxer movement, right? And so this is causing a lot of issues in our day, especially now with the COVID vaccine, but also with kids getting measles and everything else. Diseases that were very close to being eradicated are coming back because of this one man and his issues and by falsifying these results. In our class, there's absolutely no reason to falsify your results. Your grade is never based on the numbers that you get. Your grade is based off of the effort that you put into your report, your oral report at the very end, and into your experiment. Okay, so there's absolutely no reason for you guys to falsify your results. So do not do it. I will know if you do it. I'm still here. Okay, so if you have questions about this, let me know. So the next thing we're going to talk about is an experimental design. So I talked a lot about this in the other video as well. So we're just kind of going to go over a few of the main points in this that you guys do need to know and understand and that sort of thing. So the main points in this that are going to be important, you guys need to make sure you know and understand what controlled variables are. So your controlled variables are going to be any variable except that is being tested that could have an effect on the outcome of your experiment. So in this case, we have a couple up here that are already up here for you guys. So you have sunlight, which essentially means you're going to give your rose bushes the same amount of sunlight, right? The next is going to be the amount of water. So make sure that you're giving every single rose bush the exact same amount of water each time. They're going to be in the same temperature. So one's not in a greenhouse and one's not outside. They're both in the exact same temperature. You're also going to use the same type of rose bush. So you want to use the same species of rose bush. Because if you use two totally different species, then you could end up with a very different end result. And it's going to end up not being correct. You also want to use the same type and amount of fertilizer. So this is something that should be controlled in both of your bushes as well. The next thing we're going to look at is going to be the experimental variable, which is going to be the variable that is being tested. So in this case, it's going to be our two sources of water, right? So our experimental variables are going to be the well water versus the rainwater in this case. In some cases, we do end up having to have a control treatment. This doesn't happen too often. It just depends on what type of experiment you're doing. So in this case, we do not need to have a control treatment. And the reason being is because we are comparing two different things. We are comparing well water versus rainwater. However, if we decided to look at fertilizer only, so if we decided we had two rose bushes and one was given fertilizer and one was not, our control treatment would end up being the rose bush that was not given fertilizer, okay? So you only need a control treatment if you are not comparing two different things. The next thing we're going to look at is replications. So in this case, we're looking at two rose bushes, right? So one rose bush is given well water, one is given rainwater. And this is going to end up being one replication, okay? So we only have one replication in this case. A replication is just going to be the use of multiple trials of the treatment, okay? It's a really great way to increase your certainty and reduce the effects of random chance. So one great thing that we could do with this one, currently we have one replication, but if we ended up with 6 bushes, then we would end up with 3 replications, okay? So that's an easy way to remember it. So we have 6 bushes, we end up with 3 replications, okay? Replications are always super important to do. You want to do as many as possible. The next thing we're going to look at is an assumption. So an assumption is going to be anytime you assume a factor has no bearing on the outcome of your experiment, when it does, in fact, influence your results. So a really good example of a possible assumption with this one would be if one rose bush was in a pot or of some sort, and the other one was in the ground. Some would assume that this does not have any effect on your rose bushes when, however, you don't really know, right? We have no idea if it would have an effect or not. And so assumptions are very important to control as best as we can. We need to make these a control variable rather than an assumption. The next important thing for us to look at is going to be independent and dependent variables. So your independent variable is going to be your experimental variable. So in this case, our experimental variable or our independent variable is going to be the water, okay? So our independent variable is going to be water, okay? And then our dependent variable is going to be the result. So our dependent variable is going to be the growth. Oops. So it's going to be the growth of our rose bushes. So within that, we do also need to state how we're going to measure these rose bushes? Are we going to measure the height of their growth? Are we going to measure how many blooms they have or how many leaves they have? It's very important to make sure you specify all of these different things. So this is experimental design within one little thing. If you have questions and need clarification, please email me, guys. I know that this is not the ideal way to learn all of this, but I'm here to help you guys, okay? So moving on, we are going to, typically, we would design our own experiment in class. It's a pretty neat experiment. It doesn't take a whole lot of time, but because we're obviously not in class, we're not going to do it. And if I had cockroaches at my apartment, I would do it for you and show you a little video, but I don't have cockroaches at my apartment for multiple reasons, but mostly because my cat likes to knock over their container. He does not like the cockroaches. Anyway, so we're going to design a little experiment, and what we're going to do is we're going to see if males or females are longer. So our experiment is going to have males versus females. So because we have two things that are being compared, we do not need a control treatment, right? So we're going to compare males and females and their length. We're going to use a string. Well, normally we would use a string, measure our cockroach from the very tip of their thorax to the very end of their abdomen, and then measure that part with a ruler. We would get that in millimeters, and we would be able to see which is longer. So I've already done this for you guys. It's based off of the results from group A. So they are real results. I didn't falsify anything. I'm just giving it to you guys for this. However, in your lab manual, you do have a couple of questions, and we're going to go over those real quick before we move on. So on page 12, the very first one I already answered for you. So the very first question says, can you think of any examples of scientific misconduct? So we talked about the whole movement with anti-vaccines and that vaccinations cause autism. So that's a really good example of that. Number 2, so list all the controlled variables you can come up with for this experiment. I suggest you guys do this. I suggest you get some practice in of listing out controlled variables and that sort of thing. I can look at these when we get back to class in person. Number 3, do you have a control treatment? And if so, why or why not? So as we just talked about, we're doing males versus females. So this means that we're comparing the two, which means we do not need a control treatment, right? Number 4, so how many replications are there in each treatment? So we have a total of 6 cockroaches, okay? So we have 6 MHEs in our class, 3 males and 3 females. So this means that we're going to have 3 replications, okay? So we'll have 3 replications for this experiment. All right, so we're going to get into our experiment a little bit. I have everything written out for you guys. You should write all of this down in your miniature lab notebook. So your miniature lab notebook, these are very important things for you guys to remember and put down in there, okay? So you have to always use ink. If you don't use ink, then I have to take off points. I don't want to do that. So make sure you're using ink. You should also record as you work. So I know this video is a little different, but from now after this, record as you work. So don't wait till the very end of lab and have to write everything down and try and remember what you did. Record as you go along. It'll make things a lot easier. Do not leave blank spaces as best you can. So a lot of these give you a lot of space for people that write small. You end up with a lot of blank space. I write really large, and so I end up with no blank space, which is fun. But it gives you a lot of space. And in this case, you're going to do a slash. So say you have all this, you're going to do a slash, and you're going to put the date on that slash, okay? So just whatever the date is that day. My recommendation with this is to wait to the very end of class, and this is just so that you don't end up doing this prematurely and end up having to write over it, because you don't want to do that, and I don't want to grade that. So I have an example of this later on. You're also going to always date your each page. There should be a spot for it. Date the page, whatever the day is. For this one, you can put whatever day you watch this video. It doesn't matter the date. You're also going to sign it. So it's your signature, not mine, your signature at the very bottom. So make sure you do this. You should also not remove pages, so do not remove your pages. I will know because I have to grade it and I know what's supposed to be where. So don't remove your pages, and I don't want to have to take off points because you accidentally removed a page. Okay, so make sure you keep them all where they are and in order. So here is an example of what your lab 2-3 looks like in your lab notebook. I have what you're going to write down in there as well for you guys. So should be super easy for you guys to follow along. You can essentially copy this exactly as it is. So write the date. My date is wrong. Put the date that you watch this video. You also have your objectives. So we're using this with our experiment that we just talked about. So we're going to determine if adult male MHCs are longer than adult female MHCs. A possible hypothesis for this would be if male MHCs rely on aggression to compete for resources and bigger size is an advantage, then the average adult male MHC will be longer than the average adult female. So with our hypothesis, for you guys, I suggest using an if-then statement. And this is mostly just so that you guys can get used to a hypothesis and what all it needs. Within your hypothesis, though, you need to have a reason. So you can't just say males are longer than females. You have to give me a reason as to why you think this. So in this case, we have they rely on aggression to compete for resources and bigger size is an advantage, okay? So that's a reasoning for why we think this. You're then going to write out your procedure. So when you're writing your procedure, I suggest keeping in mind that if you took your miniature lab notebook and you gave it to a middle schooler that's in a science class, they should be able to redo this experiment, okay? So it doesn't have to be so in such small words that a really young person can read it, but it still needs to be in a way that anybody could replicate this, okay? So for this procedure, the very first thing we're going to do is use a piece of twine to mark the length of an adult male MHC from the forward tip of the thorax to the backward tip of the abdomen. Next, we're going to use a ruler to measure the length of the marked twine in millimeters. Repeat for an adult female MHC. Take measurements for a total of 3 male MHCs and 3 female MHCs, and then you're going to record your results, okay? So I have our results here. So these are, again, results from group A. So male 1 is going to be 58 millimeters, and then 63 millimeters, and then 60 millimeters. Our females are going to be 51, 55, and 60 millimeters. So if we just kind of eyeball this data, it looks like our males might actually be a bit larger, right? But we're going to do some stuff in a minute to figure out if that's true or not. You're then going to put your signature at the bottom. So here's an example of what it will look like if you have a lot of blank spaces. Again, you'll put the date that you do this, not this one, but this is just an example for you guys. So, like I said, we're going to get into some actual data analysis, and so we're going to do this now. You guys can go ahead and turn to page chapter 13 in your book. This is around page 16 or so. We're going to talk a little bit about the t-test. So I talked a lot about the t-test earlier in the other video. So if you have more questions, go back to it. However, the good news is you don't have to actually do the t-test longhand, okay? We have this really nifty website for you guys that you guys can look at and do the t-test through that, okay? So don't panic. You don't have to do a whole lot of math. But you do have to understand the t-test and what it's about. So we have, within the t-test, we have our numerator, which is going to be the top part, right? And this is going to compare your means. So our means are going to be this X bar and Y bar. So X bar is going to be the mean or the average of the male cockroaches, and Y bar would end up being the average of the females, okay? You then have your denominator, which is going to be the bottom part. And so your denominator is going to account for sample size. So how many cockroaches do we use? It's going to account for variability, which variance is going to be the difference in your cockroaches. So say we have some male cockroaches that are really, really big and really, really small, but not a whole lot in the middle. But all of our female cockroaches are about in that middle area. And so this is going to account for our variability. The denominator is also going to make it so that outliers have less effect if the sample is large. So what this means is if you have all of your cockroaches are around somewhere in the 60s and you have some random cockroach that is like 80 millimeters, this is going to make it so it's not as big of a deal. So with the student t-test, so what we're going to actually do with this is I have a link for you guys to do the t-test for you. So this link, all you have to do is have the numbers and input those numbers, okay? Super, super easy. Makes it a lot easier for you guys to do. It means you don't have to remember the t-test and you don't have to calculate it yourself. So we're going to go and we're going to actually do this. If I can, there we go. All right. So I got my link working and everything. So you have up here. So we have treatment 1. So we're going to make treatment 1 our males. So our males were 58, 60, and I think it was 63. Oops. Hold on. And 63. So that's what our males are. Our females were, I think, 60, 55, and 51, I believe is what our females were. So now we have both of our treatments in here. We're going to use a significance level of 0.05. This is so that we have a 95% certainty that this is not due to random chance. And we're also going to use a one-tailed t-test. And I will talk more about this in a second. So we're going to calculate our t and p values. It gives you a whole bunch of numbers and a whole bunch of stuff. I don't want to see any of this, okay? So when you guys do your own t-test, I don't want to see any of this stuff. Just scroll past it. This part at the bottom, this is what I want to see. This is what is important, okay? So it shows you your t-value, which is down here. So your t-value is going to be 1.67705, okay? So this t-value is what you're going to put. Make sure you put all the numbers, okay? I don't want to see just 1.6. I want to see all of the numbers. Your p-value is going to be 0.084419, okay? So this is going to be your p-value in this case. And we can also see that our result is not significant at p 0.05, okay? So this is not going to be significant in this case. So because of this, we are going to have to possibly refine our hypothesis. So we're never stuck at our hypothesis. And because of this, we can actually change it up a little bit. So your directionality or your tails, so one tail or two tail, is another way that you can change this up. So one-tailed is going to be directionality, which essentially just means that you have stated that one of your treatments is going to be different than the other. You're stating that one is bigger or smaller or more or less or higher or lower than the other one is. Whereas with a two-tailed t-test, you're stating that there is a difference, but you don't really know what it is yet. So you're saying that one is probably bigger or smaller, more or less, higher or lower, but you don't know what it is. This does provide a more stringent test for P. So sometimes if your t-test does not work for a one-tailed, it sometimes will work for a two-tailed, okay? So keep that in mind. But for documentation, we are going to look at it in here as well. So one thing that we do need to do before we move on is you guys actually need to calculate your degrees of freedom. So your degrees of freedom is fairly easy to do. Your formula is on page 18 in your lab manual. Very, very easy. So V is equal to the sample size of X minus 1 plus the sample size of Y minus 1, okay? So your sample size is going to be how many cockroaches. So in this case, we use 3 cockroaches for X and 3 cockroaches for Y. So we end up with a degrees of freedom of 4, okay? So super important, probably the hardest math you're going to have to do in this class, okay? So that is also the answer to number 4 on page 19. So we're going to go through the documentation for this portion of lab 3 for you guys. So you're going to put the date like you always should, and then you're going to refine our hypothesis because the first one did not work. So our second hypothesis is going to be if MHCs display multiple forms of sexual dimorphism, then male and female MHCs will be different lengths. We then have our values up here. So this t-value is actually wrong. So we're going to put in our correct t-value, which is going to be 1.67705. It's going to be our actual t-value, okay? And then we know that our p is not significant at 0.05, right? So in this case, when refining this and using our second hypothesis, we did find that p is significant at about 50%, which isn't great. So we don't really like this one. In all honesty, if we did more cockroaches, we probably could get a better sample size, or better t-test. So you're then going to write a conclusion. And so this is a really good example of what your conclusion should look like. So it says the data does not support the hypotheses. This result suggests that there is no significant difference between the length of male MHCs compared to female MHCs. The conclusiveness of these results could be strengthened with a larger sample size. So pretty good conclusion right here. And then you're going to add in your signature as well. So real quick before we move on, I do want to go over some of the questions with you guys for page 18. So the very first one is, in the t-test numerator, what could yield a larger t-value? So we looked at our, I'm going to skip this. I don't even need this. Never mind. I need to edit this out. Okay, so the next thing we're going to talk about is going to be scientific literature. So scientific literature is very important for you guys to be able to know and understand what is going to be actual scientific literature and what's some random person with no science degree just typing on a blog. So the very first type of scientific literature we're going to talk about is going to be primary. So this is going to report an investigation conducted by the authors. So what this essentially means is if we had performed our experiment and everything looked great with it and we got a lot of numbers and everything else, we could write a whole report on it and we could send it in to be published. And it would be primary because we're the ones that conducted this experiment. These are some examples of really great journals. To get published in these mean you're essentially like the best. But Cell, Science, and Nature especially are really great for looking at primary articles. The next we're going to look at is going to be secondary or review articles. So these are going to summarize primary literature on a particular topic. So essentially what this means is if I'd given you your little topic for your Lab 3 homework and I told you instead of doing the citations like you're supposed to, you're actually going to do an entire paper on your topic and you're going to compile a whole bunch of research from primary articles, and you give me this paper and it's incredible and I say, let's publish this. And so you turn it in and that's going to end up being a review article because you went through and you looked at all sorts of different things that other people had already published to compile data together. So that would be a example of secondary or a review article, okay? So this leads us to citations. Citations are super important in this class. You do need to know how to write a citation. In this class, we have a very specific way of writing a citation, okay? So you do have to do it this way. You cannot use EasyBib or Citation Machine or any of those other websites that I don't know about. Because if you don't follow this way specifically, I have to take off points and I will know if you did not use this specific way, okay? So pretty easy. You have author or authors, date, article title, journal title, volume, issue, location. Very easy to do. There is an example on page 21 for you guys. There's lots of examples for you guys to do. There's one with one author, one with multiple authors. There's one with 10 authors, okay? Lots of different ways for you guys to do this. What you're going to do is you're going to actually use Google Scholar to find your articles. You can also go to the TTU website for the library and they can also find it for you. So this leads into your Lab 3 homework. So your Lab 3 homework is not too bad, I don't think. So you're going to have 3 articles. So you have to find 3 articles and you're going to create a citation for all of these articles, a summary for each article. Your summary should be 4 to 6 sentences, okay? Do not copy the abstract. I will know if you copy the abstract. You also need to say if it's review or primary. Okay, so you need to tell me which of these your article is. You also need the first page of this article attached, okay? Because you are turning this into Blackboard, it might be easier for you to just attach the entire article that you've done. Totally fine, as long, I just need to see the first page, okay? So whatever is easiest on you guys, not a big deal. And just a reminder, this is due on Sunday, February 21st by 11 p.m. Okay, so this is due by 11. The deadline is not midnight, it is 11 p.m. Okay, so this is your lab 3 homework. You're going to use the topic that I gave you guys for this. If you don't remember your topic, go back to the very first slide and pick one of those 3. Okay, so you guys are now going to be given the opportunity to design your own experiment. We're going to do this. We'll talk a lot more about this when I see you guys in person again, but you are also going to end up contacting your lab partner. Hopefully you guys got their contact information to work with them on this. So this experiment is going to occur over the next 4 weeks. So we only meet 4 more times in this class, and you guys are going to work on this experiment for the first 10 minutes of every single class from now until the end of the semester, okay? Your experiment has to end up with quantitative data, okay? You have to give me quantitative data when it comes to this. So remember, quantitative data means that you have to give me numbers. You also have to have 2 treatments. So you cannot have a controlled treatment. You have to have 2 treatments, which means you have to compare something. You also have to have only 1 experimental variable. So you cannot do multiple experimental variables. You can only do 1 thing, okay? All other variables will have to be controlled. You will need as many replications as possible. So at the very least, you should have 4 replications. However, you should have way more than 4 replications. We have plenty of cockroaches for you guys to use, and you have the first 10 minutes. Most of the time, it will not take you 10 full minutes to do 1 replication, okay? So do as many as you can. You also have to have my approval, which essentially is just so that I can make sure that you guys are doing what you need to do, and you're going to get the best results as possible with this, and you can do an oral report with this later on. So these are the rules and regulations for your experiment. This is for your long-term experiment, which will be your oral report at the very end, okay? So since you guys have never done this before, I do have some questions that you guys can do. So these are going to be the 6 research questions that you're going to choose from, okay? So you have any of these that you can pick from. So the first one is, are MHCs motivated more by fresh or dry food? Are male or female MHCs faster? Are flipped MHCs able to write faster on a smooth or rough surface? Do MHCs move faster in dark or lit conditions? Do sounds influence the ability of MHCs to complete a maze? And are male MHCs more prone to right or left antenna breakage? So you will need to work with your lab partner, and you'll figure out which of these research questions you want to do, okay? You'll then need to come up with a hypothesis and a procedure for all of this as well. I will have to check your hypothesis. This is so that I can make sure that you are coming up with a correct hypothesis and everything like that. This leads into your long-term experimental design worksheet. So this is something you're going to turn in. The due date for this is going to be February 26th, which is going to be a Friday. So I've given you as much time as possible to do this. Because of the weather and everything. So you're going to get with your partner. You can get with them via Zoom. You can turn this into a Google Docs. I don't care, just as long as you guys get it done. But I do have this as an example, and we're going to use the experiment we did for class, which is going to be if males or females are longer. So the very first thing you're going to do is you're going to state your research question. So are male Madagascar hissing cockroaches longer than females of the same species? What are the possible explanations for this observation? Males are aggressive and females are not. Develop a hypothesis. So I want you to write your hypothesis here, okay? And this is so that I can check your hypothesis and make sure that it is correct well before class starts, okay? So make sure you actually give me your hypothesis here. I will count off points if you don't put it here, okay? So please, please, please put it here. if it's incorrect, I won't count off points. I just want to see it, okay? I need to help you guys as much as possible. So 3A, state your explanation for this phenomenon. Male Madagascar hissing cockroaches rely on aggression to compete for resources, and bigger size is an advantage. B, state one prediction that can be made from your prediction. And the average adult male Madagascar hissing cockroach will be longer than the average adult female. So number 4, describe the different groups or treatments in your experiment. Be as specific as possible about any conditions or experimental variable that will be changed between the groups. So the treatments are going to be adult male MHCs and adult female MHCs. Your experimental variable is the difference between the two treatments is going to be sex, okay? So B, how many cockroaches will you use for each group or treatment? Each treatment will have 2 Madagascar hissing cockroaches. So it'll have 1 male and 1 female, okay? So this is why they will have 2 in this one. And then why do you choose the number of cockroaches? While 2 trials are an inadequate number to accurately represent the MHC population, my section only has 4 cockroaches, and my TA has not authorized us to permission to obtain additional cockroaches for this experiment. The results are more likely to be affected by outliers or chance. So in our case, we actually have 3. So our class has 3 cockroaches, or 3 of each sex. So we have 6 total. We can also utilize the cockroaches that are in the colony. So you cannot actually use this for your answer, okay? So keep that in mind. Do not use this as your answer. Come up with your own, because you will be able to use more cockroaches. Next, so what variable will you measure? So the measured variable will be the length of the Madagascar hissing cockroach. How will you measure this variable? You need to be as specific as possible. So we will use a piece of twine to mark the forward tip of the thorax to the backward tip of the abdomen along the curve of the MHC's back. A ruler is used to measure the length of the marked twine in millimeters. 5B, explain 3 variables you will keep constant in your experiment. These are going to be your controlled variables. And explain why you need to control these variables. So we need to control the species, developmental stage, and measurement equipment / technique to control all the trials of both treatments. So you also need to explain why you are stating these things, okay? So explain to me why each of these needs to be controlled. The next thing is going to be, should you repeat your experiment? Why or why not? Your experiment should be repeated to increase confidence in the results by reducing the impact of uncontrolled variables and random chance. This part at the end does not pertain to you, because you will do this multiple times, okay? This is, again, for the one that we would have done in class. So you will actually do as many replications as possible. You should have at least 4, but you should do more. Number 7, what are the independent and dependent variables in your experiment? So your independent variable is going to be the sex of the cockroach, and your dependent variable is going to be the length. And finally, we have the last page, and so you have to graph what you would expect to observe if your results supported your hypothesis. And you have to make sure you label both axes and include the units. So this is what this one would look like. We then have males versus females, okay? So this is going to be what our graph would most likely look like. Then we have number 9, describe what conclusion you could make if your results supported your hypothesis. So within this population, male Madagascar hissing cockroaches are significantly longer than female Madagascar hissing cockroaches, which supports a size advantage for aggressiveness. And then number 10, describe what conclusion you could make if your results did not support your hypothesis. There is no significant difference in the length of male and female Madagascar hissing cockroaches, so aggressive behavior has no effect on size within this population. So this example can be found on Blackboard for you guys. It's all in the Lab 2-3 section. You guys need to make sure that you complete this. You have to complete this with your lab partner, okay? If you're not getting a response from your lab partner, then you can do it by yourself. Because you do need to get the points. It's worth 20 points, so it does give you a good bit. So make sure you complete this. You have to complete this before you can start your experiment, okay? So make sure you do this as soon as possible. Again, if you have questions, if you need anything, please email me. And I really hope that you guys have all been safe through all of this and haven't had too much issues with the cold or not having water or not having electricity and everything. I hope you guys have been safe, and all of your family members that are probably in various parts of Texas have been safe as well. So if you have questions, please email me, guys. All right. Hope you have enjoyed your essentially a spring break. So see you guys later.